Let's take a look at some of the X-linked traits in humans that, like the fruit fly eye color, have different patterns of inheritance that can be seen in males versus females. Now, these are all considered disorders caused by something that's different from what is considered normal. And they are also caused by recessive alleles linked on the X chromosome. So these include color blindness, muscular dystrophy, and hemophilia. And if you haven't heard of these, I'm sure you've heard of at least the first one. We're going to go through a little brief introduction to each one. First one being color blindness. The cause of color blindness is abnormal cones on the retina of the eye. These are color receptors that transmit um, light information of the visible light spectrum for color from your retina, where these cells are, to the eye through the optic nerve. So there would have to be some kind of a mutation in the gene that encodes for the cones. And so the brain of a person who's colorblind is getting faulty information or missing information. So quick check, can you see the number here on the slide? Hopefully you can. If not, maybe you already know that you are colorblind. There's different kinds of color blindness. It really depends on which cones are abnormal or missing. Red-green color blindness is the most abundant in the population. This is not red-green. You should see the number 26. I don't think I've met a student yet who couldn't see this one, but I'm not doing the full colorblind test. So don't just go by this, but that's basically one of the tests that somebody would have to do to determine if they're colorblind, is whether they could see that number there. All right, so that's color blindness. Let's take a look at a more concerning disorder, because color blindness, somebody can live a pretty normal life with that, but having muscular dystrophy would be a bit more of a challenge for somebody, because this is a disease that's characterized by a progressive weakening of the muscles and loss of coordination. So throughout the course of their life, it just becomes more challenging for them to do bodily movements, walking without the aid of crutches. They might need a wheelchair. So this disease affects about 1 in every 3,500 males born in the United States, and these males don't typically live past their early 20s. So it is a very serious disease, and it's not curable. There are treatments somebody can get for this to just help treat the symptoms, but there is no cure for muscular dystrophy. And similar to that, there's no cure for this third type of sex-linked recessive disorder called hemophilia. And this is caused by an absence or abnormality of one of the proteins that helps the blood clot. So people that are hemophiliacs, they could actually suffer from, if they got a cut, they could bleed out if they don't get care for that wound. Their blood can't clot itself to protect them from losing blood. And also, if they bump into something pretty hard and get a bruise, they could have internal bleeding that can lead to other issues: pain, swelling, muscle wasting, which is when the muscle basically starts to degrade inside the body. And that can lead to other issues: chronic pain, limited mobility. So it could be fatal if the injury is severe enough that they lose blood or have internal bleeding and all these issues. But it's not as serious, I guess, as if you could compare them. Muscular dystrophy. And like muscular dystrophy, there's no cure, but there are treatments. Somebody could get injections to supplement their body with that missing protein. So these diseases are, or disorders rather, are found in the human population, both in females and in males, but they are more abundant in males than females. And it has to do with the fact that it's X-linked and how X-linked recessive traits manifest themselves in males and females because of the genotype for male and female. So to have a better understanding of that, we're going to be doing some Punnett squares. And we'll do one together, and then you have some practice problems that you can work on in your course pack worksheet. And I'll direct you to that when we're ready. But these are some of the guidelines and rules and just some new terminology regarding doing Punnett squares and making predictions about the crosses for a X-linked recessive trait. So just in general, X-linked alleles are passed from, if it's the father that has the X-linked recessive trait, he's going to pass it to all of his daughters, but none of his sons. You'll see why again through the Punnett squares. Whereas a mother is going to pass her X-linked alleles to both her sons and daughters, since she has two X chromosomes. If a female is heterozygous and has one recessive allele, and that is the allele for the trait, and a dominant allele that's normal, then she's going to be called a carrier for that trait. And that's not a new term. We've had that before. When it's a disease and they're heterozygous, they would be considered a carrier. And a male who has, so male would be XY. There's really no such thing as heterozygous for a male, because if it's an X-linked or sex-linked trait, he only has one allele for that, because it's either X or Y linked. So there's a new term for a male. When he has the recessive allele, he does express the trait, and he's called hemizygous, because there's no dominant allele to mask the recessive allele. So whereas a female who only has one recessive allele will not express the trait, she'll be still normal. A male only needs that one recessive allele to show the trait, or the disorder in this case. This is part of the reason why males, you see them more in males than in females, these disorders. So let's do an example of a Punnett square for one of these types of problems. Okay, here's our Punnett square scenario. We're going to show how a colorblind daughter could be born to a colorblind father whose wife is a carrier. So let's start with identifying the parental genotypes. The colorblind father, well, he has to be XY for his chromosomes, for his sex chromosomes to be a male. And since he is colorblind, he has the recessive allele for colorblindness linked to his X chromosome. So his genotype is X superscript little n for, the recessive allele. I'm using capital N for normal and lowercase n for the recessive allele for colorblindness. And I'll add a key to the slide in a minute for that. And then he has the Y chromosome, which there's no allele for colorblindness because it's not associated with the Y chromosome. The mother, well, she is a carrier, so that means she doesn't have colorblindness expressed, but she has the allele for colorblindness. She's carrying it. So her genotype for being a female is 2X chromosomes, but it's X superscript capital N, a dominant allele, X superscript lowercase n, a recessive allele, making her the carrier. All right, so see if you can set up the Punnett square, and I'll do that as well. You might want to go ahead and pause the video while you're working on that. So I went ahead and added a key on the left side to show what I'm using for the alleles, in addition to the X and Y chromosomes, which you need to know and means that it's male or female, of course. And so I also solved the Punnett square here, and make sure that you're getting the same as me, and we can go through that together and make sure that we are. The first way I want to interpret the results is just doing an overall phenotypic results, but then we're going to break it down into males and females, because sometimes when people have kids, they find out first if it's a male or a female, and then they want to know whether that child has a genetic disease. But overall, there is a 1 in 4 chance of having this is the first box, a normal seeing girl. That's this one here. It's the only probability of a normal seeing girl. Now, let's finish up the girls. Actually, let's read it like we usually do from left to right across the top, just for consistency with how I've done it in the past. So the next box shows a boy, XY, with also the dominant allele. So that's a normal seeing boy. Okay, now to the bottom left, this is a girl that's colorblind because she has two recessive alleles, making her colorblind. And lastly, we have a boy who is colorblind. So there's a 25% chance or 1 in 4 probability of a colorblind boy. Okay, so that's an overall breakdown. If you look at every single outcome and what their unique phenotype would be. But there's another way to look at this as well, because, um, personally, when I found out I was going to have a child, I wanted to know, I'm a planner, I wanted to know if it was going to be a girl or a boy. And so that was the first information that we got. And then, of course, regardless of the gender, you want to know if they're going to be healthy. So if it's a girl, then we're just going to look at the boxes that have that 2X chromosomes. So just here now in the pink, there's only 2 possible outcomes for girls. So the denominator is going to change based on that to be out of 2. So there's a 50% chance or 1 out of 2 chance that she will be colorblind or normal. I need a larger box here. Okay, so there's the summary for if it's a girl, we have a 1 out of 2 chance of her being a normal seeing girl and a 1 out of 2 chance of her being colorblind. We can do the same thing for the boy. If we find out that the child is going to be a boy, then it's actually the same. 1 out of 2 chance that he is normal seeing and 1 out of 2 chance that he's colorblind. So back to the original question, the prompt, the instructions were to perform a Punnett square to show how a colorblind daughter may be born to a colorblind father whose wife is a carrier. So were we successful in doing that? Did we show how there could be a colorblind daughter? Yes, we did. In fact, there's a 25% chance that their child will be a colorblind girl. So now I'd like to encourage you to try some additional Punnett square practice problems for this type of inheritance, and you'll find that in your course packet if you have the printed version with the page numbers. It is pages 40 and 41, or you'll find it in the Mendelian genetics section, just after the Mendelian genetics section in the course packet. The answer key will be posted on D2L when appropriate for your class.